Hello, my name is Martin. I'm working mainly on the Unicorn and the BCI core systems. And this is Sebastian, my colleague, which is doing signal processing and gives us a lot of important research insights into the system. Today I'm going to present the Unicorn Hybrid Black and the Unicorn BCI Core-8. And I've already set up Sebastian as a subject with the Unicorn Hybrid Black. On the slide, you can already see the Unicorn Hybrid Black. It's a system with 8 electrodes, and it's more prone in the direction of consumer brain-computer interfaces. We have 2 different versions of the Unicorn Hybrid Black. So one is a ready-to-use device, and the other one is constructed for makers as a PCB version, so you can build your own headsets and build the PCBs into your own mechanical headset. As you can see, we have 8 electrodes, and we have an electrode stand for 8 electrodes, and we have this disposable sticky electrodes that usually go to the mastoideus. Here you can see the 8 hybrid electrodes assembled to the amplifier. So in the previous slide, you can see the electrode clips, and you can simply slide the electrodes on. And you can see that there's a little hole in the middle of the electrode. That's how you can use the electrodes with gel. You can insert gel in there. So these electrodes are made of carbon rubber, and they can be used with gel or in a dry setup. So here you can see it in a more detailed view. You have the hybrid electrodes, and you can see the pin. The pins are there so you can get through the hair and touch the scalp directly, because it's very important that you have a good contact to the scalp, because that's where the voltage actually originates, and you're just picking it up with the electrode. So it's important that you're touching the scalp with the electrode. And if you still are not confident with the EEG signal, you can add gel and improve the contact in between the skin and the electrode a bit more. So with these hybrid electrodes, you can create dry setups or gel setups. So it should be feasible for all of your research projects. The Unicorn Hybrid Black has 8 channels, as mentioned already, and an amplitude resolution of 24 bits for a voltage range of plus minus 750 millivolts. So therefore, you get a very high accuracy for very small voltages, and that's what you need, because EEG is usually occurring in an amplitude range of plus minus 50 to plus minus 100 microvolts. So you want to zoom in with a very high precision, and therefore we have a 24-bit ADC built into the device. To improve the signal additionally, we have a very high oversampling rate that improves the signal to noise ratio. As mentioned already, we have these hybrid and dry electrodes, and we've already pre-assembled them at locations that you would need for BCI experiments. So we have 1 electrode in the front, if you want to use the frontal lobes. We have 3 electrodes over the motor area, so you could do motor imagery experiments. And then we have 4 electrodes at the back of the head, so you can do visual EEG experiments. So that's the hardware setup, and additionally to the customizable hardware, we have a lot of software tools to support research. So we deploy the software via the Unicorn suite. And the Unicorn suite is more or less an app store where you get applications and development tools. For the applications, we have some tools that are included in the Unicorn suite and are available for free, and some of them require a license that can be purchased in the g.tec web store. So to give you an overview, we have the Unicorn recorder, That's one of the applications we're going to see in a couple of minutes. In the Unicorn recorder, you can basically visualize EEG, you can pre-process EEG, you can record EEG, and you can forward it via lab streaming layer or UDP. Then we have the Unicorn Speller. This is a P300 application. As the name already mentions, it's a speller. The people can select letters and write words just using their thoughts. We have the Unicorn Painting. This is an application that was originally developed for ALS patients because a lot of ALS patients wanted to create paintings and stay creative. And with this tool, they can still create digital art just using their thoughts. We have the Unicorn g.tec, which is a neuromarketing application. That's also one of the applications we're going to show later on. So it's basically presenting images and triggering interest in your brain or not. And we're going to see how the BCI is detecting whether you're focusing or attracted by an image or not. And we have the Unicorn band power. So a lot of people know that EEG has different frequency ranges, from delta waves, which are occurring by sleep. They have a big amplitude and very slow frequency, up to beta or gamma activity that are very task related and occur at higher frequencies. And with the Unicorn band power, you can detect delta, theta, alpha, beta, and gamma waves and measure them. You can also forward it to an external application if you want to build, for instance, a neurofeedback application. And then we have two games, a platformer and a puzzle game, and the brain body game, which are also included in the Unicorn suite and are available for free. These are the applications. They are provided in a compiled form and can be started out of the box. But a lot of users are researchers or developers or makers, and therefore we also have a lot of development tools for different programming language. So we have an API for C, for C# or .NET, for Python, for Simulink. And if you want to go to the network, we have an interface for lab streaming layer or UDP. If you're a game developer, we are also providing an interface for Unity. That's a game engine. We've also used for the free games. It's also included in the Unicorn suite Hybrid Black. And for the makers and developers, we also have a lot of tools on GitHub. So if you're participating in the hackathon, we also have all of the software manuals on GitHub. So if you're looking for some information on how to use the software, you should find that on our GitHub. And we also have some open source tools like the UDP interface or the raw data interface for Unity, a Linux C API, some datasets, and so on. So you can also take a look at that if something is interesting for your project if you're using the Unicorn Hybrid Black. And now I'll come to the first demo, and we're going to start with a simple signal quality check. And I've already opened the Unicorn suite Hybrid Black. You can see we're in the app sections where we have all of the tools that I've mentioned previously. We have all the development tools. I've mentioned that there are some software modules that require a license key. So if you purchase the license key, you can insert it in the Unicorn suite and activate your software module. So if you're, for instance, activating the Blondie check, it will be listed in here. And the button, which is default locked, is disabled, and you can open the software. In the My Unicorn section, you can discover and pair with devices. You can also check connection state if you're able to connect with your Unicorn and check the Bluetooth environment. So in best case, all of these sections are green. And if you're looking for documentation or the manuals, there's this question mark sign that is directly leading you to the page where you're finding information about all the software modules, about the hardware, and so on. Now we're going to start with the Unicorn recorder and signal quality of the Unicorn Hybrid Black. We've already assembled the device to Sebastian, and we have 2 electrodes at the back of the ears at the mastoideus. This is a bony structure where you can find very little electrical activity, so we use that as a reference electrode. And then we have the 8 electrodes assembled, and 4 of them, the ones in the back of the head, channel 5, 6, 7, and 8, are gelled, and 4 are not gelled. So we can see the difference between dry and wet electrodes. So EG is usually located in an amplitude range of ±50 to ±100 microvolts. That's why we set scaling to this range. And by default, if we're using surface EG that is non-invasive, we often use a frequency range of 0.5 to 31 to 30 hertz, and a not filter that removes the power line hum. And I can show you what the EG looks like if we do not apply a power line hum. Then we can see a lot of noise, especially on the 4 electrodes that are used in a dry setup. And as soon as we apply a notch filter, for instance, a cascading notch filter, we can see that this noise is removed perfectly. So now the signals immediately look good. And then we can also see a difference in between the gel and dry electrodes. So if Sebastian is not moving or not doing anything, the signals almost look identical. So all of the signals are very flat and look good, and we can do some tests to check whether we're really looking at EG or not. So Sebastian, can you blink a couple of times? So we can see eye blinks on all of the channels, especially on the front channel, channel 1. We can see that the amplitude is very big. Then you can clench your teeth a couple of times. So these are EMG artifacts from the chins that you can still see in the EEG. And you can close your eyes for about 10 seconds. And now we can see alpha activity, and that's actually coming from the brain. All of the other signals that we've seen so far are artifacts. And the simplest way to trigger alpha activity is closing the eyes, because then your visual cortex relaxes and you can the neurons are synchronizing. You can see a signal with a frequency of around 10 hertz that is bigger in amplitude than the baseline. That's what we're seeing here very nicely. So can you open your eyes again? Now we can see baseline activity, and you can close your eyes again. Now we can see this sinus sine signal, and we can see alpha waves with a frequency of 10 hertz. And as mentioned, we've gelled 4 electrodes and we left 4 electrodes non-gelled. And you can see the differences not only in the power line hum that is acquired. So dry electrodes work perfectly if you're sitting as still as Sebastian is right now and nobody's moving around. But as soon as I start moving around Sebastian and introducing artifacts, I can easily disturb the dry electrodes, but the gel electrodes remain very stable. If I start walking next to Sebastian, you can also see that on the dry electrodes. So if you're using a dry setup, you want to sit very still and calm, not move too much, and check that you're also quiet. If you're in a more crowded environment, or if you're moving a bit, you want to add some gel. Now I'm adding the gel to the other 4 electrodes, so we can have a stable experiment while I'm doing this. Added a bit gel. It takes a little bit of time until they settle, but you can see I can use around the vehicle beforehand. Okay. That's how we usually check the signal quality. And I also mentioned that we've set up filters. I already showed the impact of the notch filter. And I can also show you the impact of the other filter. If we're not applying any bandpass filter, the signal will be floating around. So for research, you often want to use raw EEG, but then you have to deal with a couple of artifacts, like you have a DC offset that you might want to remove or not. As soon as we add a high-pass filter, the DC offset is removed, so we can add a high-pass filter of 0.5 Hertz. Now you can see that the signal is starting to float around plus minus 100 microvolts. And we can add a notch filter, so we remove all the power line hum. And the signal looks more clean again. And it's still a bit noisy. Why we usually add a bandpass filter from 0.5 to 30 Hertz, that we remove the high frequency noise again. And that's usually what we are using for surface EEG setups, because for surface EEG, most of the signal is located in between 0.5 to 30 or 50 Hertz. So that's something that you usually want to apply. And we can also show the Oscar filter. Right now you can still see we have some minor artifacts, and therefore we developed the Oscar, which is an artifact removal algorithm. And it takes about 20 seconds to settle, and then we can again try to introduce some artifacts, but Oscar is trying to recover as much signal as possible. So we have good EEG, and now Sebastian, you can try to introduce some artifacts like clenching the teeth maybe, and you can see the EEG remains very stable. You can blink a couple of times. There's still some eye blinks visible, but compared to what we've seen previously, removed pretty nicely. And you can close your eyes. And we can see alpha is still there, but a lot of artifacts are removed. And maybe you can twist your head a little bit, or move the head a bit to the left and the right. You can see EEG is very good. If you stay still and close your eyes again, alpha is still remaining. Now we can remove the Oscar again and look at the signals again. So you can blink your eyes a couple of times, and you can see the artifacts are way bigger. You can clench your teeth a couple of times. Again, the artifacts are way bigger, and you can move your head a little bit. And the EEG is still good. So that's the impact of the Oscar filter, which was developed in-house by Johannes. And the idea of this algorithm is that you're recovering as much signal as possible while removing as much artifacts as possible, which is very tricky because frequency ranges and amplitude ranges are overlapping for EEG applications often. And in the recorder, we now check the signal quality and just looked at EEG, but you can also record EEG. So we have the settings section in here. You can enable or disable additional channels, like we have an accelerometer gyroscope if you want to check motion. We have a battery level, counter, validation indicator, and a timing channel. These are also channels that you might want to record if you're doing research. In the recording section, we have a data logger for raw data, We have different file formats, so you can record raw data in a CSV format, which is a very commonly used comma-separated format. Data is recorded as a plain text, and in almost every programming language, you will find a CSV importer. So no matter if you're using Python or MATLAB or whatever, CSV is often supported. And we have a PDF logger. So PDF is a very commonly used file format in EEG research. A lot of tools like NNE are supporting this file format. So therefore, we have a raw data logger for the PDF as well. And if you're not too much into signal processing, you can already pre-process data in Unicorn Recorder, as shown before, with bandpass filters, Oscar, or notch filters, and send the process data into CSV or PDF files. And we also have network channels, so you can pre-process data in Unicorn Recorder and then forward it to your own application to do some signal processing or an application with front end or streaming it into the cloud, whatever you want. And therefore, we have a UDP interface and a lab streaming layer interface. So with UDP, you're very free in whatever you want to do, and lab streaming layer is a commonly used interface in research. So therefore, we also added raw data and process data streams for these two network interfaces. And if you're doing event-related research, then you often want to send a trigger to some application or synchronize it with your data file. And therefore, we also have a UDP input. So you can send triggers from, for instance, Paradigm Presenter that we provide into Unicorn Recorder and record the data synchronized with your trigger. So these are the capabilities of Unicorn Recorder, and we will see a demo with the network interface and file recording when we're testing the BCI core later on. So now we know that we have good EEG signal. We want to test an actual BCI application. So for now we've seen data acquisition and recording, but in a BCI application, you usually have a feedback and can select or control something with the brain. And for this test, we want to show you the Blondie check. The Blondie check is P300 or event-related potential-based application, where you're getting images presented and they trigger a P300 if they interest your brain, and don't trigger a P300 if you're not interested or paying attention to the images. Before starting the application, we again check the signal quality and everything looks good. And then we have some predefined picture sets where we have, for instance, grayscale images, which are less interesting for your brain, and an image of a blonde lady. That's also where the name originates from, because we've seen that faces attract your brain pretty much. And we have another picture set with multiple ladies versus grayscale images with different faces, logos, vacation images, whatever you want. You can select some of the predefined picture sets or create your own one. So we have this set editor where you're just dragging and popping images in, and then you are able to test it against your brain later on. So as mentioned before, all of these images that we're seeing here right now are going to be presented to Sebastian now. And images that attract his brain will trigger a P300 and other ones will not. And now it takes about 1.5 minutes of training, where Sebastian is only staring at the screen right now. And I hope you can see it. even in the stream, that the images are changing, and sometimes the blonde lady pops up and your brain is trained to detecting faces and will pay attention to the blonde lady, while the other images are not as interesting. So here right now we have about 10 images and they're flashing very fast. And after about a minute or a minute and a half of training time, the system should be ready to detect faces. And right now we've set each image to occur around 50 times. So usually you should set something in between 30 repetitions to 150. These limits should be fine enough to train a P300 classifier. And we're already done. And now we can test the system against the same picture set. And in best case, we find out that the system is able to detect whether Sebastian is looking at the grayscale image or not. And we can also check the response to different images in here. So here we're looking at the grayscale image, and you can see very little response. While if you're looking at the blonde lady, it looks much different. And that's what the system is trained to. So now we can check if it really works for the blonde lady, for Sebastian. And with 100% certainty, the system is able to detect whether Sebastian is looking at grayscale images or at the blonde lady. If you wouldn't have a good calibration signal, that would mean either your training picture set was bad or the subject didn't pay much attention. Then you could repeat the test or repeat the calibration. But since we're good, we go to the ranking, and now we can actually rank images according to the P300 response. And we will start again with the same picture set to see if it really works that Sebastian is selecting something with his brain and ranking images. So this goes very fast right now because training requires a lot of repetitions, but application is working very fast then and looks good. The blonde lady is at rank 1 and all of the others are ranked later on. And now we want to test the same experiment with more images and probably go a little higher in repetitions. And we would estimate that the face images are ranked to the front first ranges, while the others are a little bit behind. So now each image will flash 10 times. And in that case, the face images are ranked first, and the other ones are behind. And looks good. Within the first 6 images, the ladies are ranked. And interestingly, Sebastian is triggered by star images because we repeated this experiment last Friday, and also the star image was ranked 5. That looks like the system is pretty reliable. And yeah, you can use this image ranking system for all kind of applications. We had neuromarketing applications where people try to rank their logos or things they like versus they don't like, food, vacation, locations. All these kind of things should work. The only important thing is that you should create a training paradigm that fits your application paradigm. So we did a small study where we tried to compare grayscale images versus faces, and that worked perfectly. But if we apply that classifier to a picture set where you're trying to rank faces you know versus you don't know, it didn't work perfectly. But if you train to faces you know versus you don't know, then you could rank images perfectly of faces you know versus you don't know. So if you want to make a classifier for food, for instance, you would have to create the training picture set of food that you like versus you don't like, and then you can apply that classifier to your ranking system. All right. We might have some more time and make and also show a short game. So now we show some EEG signals, some research application or neuromarketing application, and we also want to show you a short game. And therefore, I'd like to show Brain Body. This is also a game based on ERP signals. And it was created during a hackathon, and the idea was to create the game for impaired children. And impaired children are not able to use their hands as they want, but they can use the brain. And some of the children had siblings, so the idea was that one is controlling the keyboard, the sibling is not impaired, the other one is using the BCI. Only if they both together they can solve the game. So the principle is similar. Sebastian is now looking at this flashing dot and is training a classifier. And later on in the game, he has to look at less flashing dots as well. And only if he can focus on the flashing dots, we can solve the puzzle together. We have a good classifier. Now I can move the keyboard, and Sebastian is changing the environment. So he looks at the dot is selected and it's changing the environment, so I can control the keyboard and continue with the game. So now I'm not able to move on, only Sebastian is the BCI. And you can already see this is working quite fast. So within about 2 to 4 repetitions, Sebastian can select something with his BCI and we can do a game. And that was created with the Unicorn Unity interface. So if you to create a game within Unity, but some tools that you can use to build similar games like this. Maybe as a hint for hackathon participants, usually create a game that is only based on BCI, it becomes boring quickly because the BCI is still rather slow compared to the keyboard. If you find people in BCI and application. So we have this game and a couple of similar games in the Unicorn suite, and you will also find a Unity interface in here. So it's basically designed to be more or less drag and drop. So you don't have to know a lot about signal processing and similar stuff. And now I'd like to go to the next device, the Unicorn BCI core. And therefore I'm sharing my slides again. Here you can see the Unicorn BCI core already. So we have this small device here, which is the amplifier, and then we have different base stations. So the amplifier is measuring the signals and is transmitting them via Bluetooth. The base station is basically the analog front end where you have different mechanical structures and caps, EEG caps to measure EEG. And you can see two of the setups that we're currently providing, one with 8 hybrid electrodes and the other one with gold cup electrodes. And you can see the amplifier in the middle. And the amplifier features some magnet as well as the docking station. So simply put the device onto the docking station to turn it on and to be ready to go. And so far we used it for a couple of different applications. Like one would be EEG for humans. So you can see the here we conducted a 300 experiment. You can see the filtered data on the bottom part in Unicorn BCI core recorder, and you can see some ERPs on the top image. But lately, EEG also often has been used for animals, and you can see some applications where they also did some ERP experiments with horses and dogs, and therefore they created apps for animals specifically, so you can fit them onto a horse's head or onto a dog head, or you can see the gold cup electrodes, which are fixed with some electrode gel. And you can again see the Unicorn BCI core, the middle head here. You can see the 8 hybrid electrodes in this case. Also to compare it to the Unicorn Hybrid Black, here are some hardware specifications. So for the Unicorn Hybrid Black, we have a Bluetooth serial port profile or RF-COM. For the Unicorn BCI Core-8, we chose Bluetooth 5, Bluetooth Low Energy as an interface. We have 8 DC coupled analog inputs. And again, we have a resolution of 24 bits because we have to zoom in to a small voltage range. So we can measure signals in a microvolt range. Also for this device, we choose a high oversampling rate to achieve a good signal-to-noise ratio. And we chose a sensitivity of plus minus 150 millivolts. We have a sampling frequency of 250 hertz. And usually we achieve around 6 hours of measurement time, or in connection mode, it lasts even a bit longer. And we also have a very high input impedance because that is important for EEG recordings in order to not lose the signals due to impedance problems. And here you can see the available base stations for Unicorn BCI Core-8. So we have different setups for 4 and 8 channels. For the Sahara soft hybrid electrodes that you've seen already with the Unicorn, these are the carbon rubber electrodes that can be used in gel and dry setups. And we have gold cup electrodes, which can be used with a special gel, only in gel setup. And for the software environment, the software for the Unicorn BCI Core-8 is deployed via g.tec Suite 2024. And we have the Unicorn BCI Core recorder in there. And similar to the Unicorn recorder, it can record CSV or PDF files. You have an UDP and LSL network interface. You can send UDP triggers there and use paradigm presenter, which is included in the HYSYS, to present stimuli. And one of the major ideas is that you using Unicorn BCI core recorder to record and precondition data and then stream it to HYSYS, which is a drag and drop editor for signal processing applications. And you can do prototypes for different kind of signal processing in there easily. And if you want to do offline processing, you can use GBS Analyze, which is also included in g.tec Suite 2024. Or if you are a Python programmer, you can use gpipe, which is a Python SDK, and that will be presented in a couple of days by my colleague Johannes Grunwald. So if you're interested in that, you should take a look at that talk. And here's some examples that we also want to show now, where we're presenting a paradigm, forwarding the triggers into Unicorn BCI core recorder, where we are aligning data and triggers. And then we want to forward data and trigger to an external application, in this case, g.HYSYS, where we want to create the P300 experiment. So this is what the setup should look like. You have paradigm presenter. This is a software included in g.HYSYS, and this is installed with the g.tec Suite 2024 and the HYSYS. Then we're sending the trigger to Unicorn BCI core recorder, where we're aligning data and trigger. Then we're sending data and trigger into g.HYSYS, and we're going to do signal processing in here and show a P300 experiment. That's what we are going to do now. And I quickly have to set up. Before starting an experiment, we want to check the signal quality. And therefore, I'm opening Unicorn BCI core recorder. I hope you can see it. Now we're connecting to the device.
This is still basically wrong.
So now we set up Sebastian, and we're measuring with 5 electrodes right now. And we can see signals floating in, looking good right now. So can you blink a couple of times? So we can see eye blinks. Can you clench your teeth a couple of times? We can see EMG artifacts. And can you close your eyes for a couple of seconds? And we can see alpha waves floating in. So since we're in a dry setup, we can easily disturb the electrodes by moving around. So I try to sit as calm as possible beside Sebastian. And EG is looking good, so we're going to set up paradigm presenter. So in paradigm presenter, we're going to load a visual ERP paradigm. And I can show you what it looks like. The paradigm starts after around 10 seconds. And you can see that triggers are sent from paradigm presenter to Unicorn BCI core recorder, and we can see the triggers arriving in here. That looks good. That's the first part of the experiment. And the second part is to stream the data onto MATLAB. And we've prepared some signal processing in here. So we're going to stream raw data via LSL to MATLAB, where we're receiving data. We're pre-processing with some filters, and then we want to show the event-related potential in here. So we're going to start the model. Signals are still transmitted. Okay. Simulink model is started. Perfect. And now we can see that we are also receiving data in HYSYS. So here we can see the process data that we are receiving from Unicorn Recorder in HYSYS, and we can also set it to a similar time and frequency range. So that looks good. And now we want to also open the ERP scope. So in here, we want to see the event-related potentials later on. And now Sebastian is going to look at the paradigm again. We can check if event-related potentials are updated in real time. So let's make it a little bit bigger so we get a good stimulus presentation. And now we have to wait for about 2.5 minutes so we can see the event-related potentials. So right now, also Sebastian is doing nothing except looking at the screen, and once a second, this image, this vacation image, is popping up, and that is triggering a visual response in his brain. And we have to repeat this process very often, so we can see event-related potentials in the average of all the trials. This is what the signal processing in the background is doing right now. Just a quick picture. Yeah, we can already see in the background that the signals are updated with every blink. So all the signal processing is running in real time in parallel right now. And in best case, we can see some good ERPs after about 2 minutes. At last 30 seconds. All right. Now we can take a look at the ERPs. And we can see, especially on the front panel, and at CZ, where we would estimate biggest P300 response, we can see at 300 milliseconds some response. We can measure some good P300s with the BCI Core-8 as a dry setup right now. That's some common BCI experiment that you might also want to do during the hackathon. All right, then I'll come to an end with my presentation. We'll be finished at 10 and make space for the next speaker.
Thank you very much for the presentation. There are a couple of Q&As. Subashini is asking if the signal-to-noise ratio is negative in dry electrodes, should we consider the signal or reject it?
Like the signal-to-noise ratio cannot be negative. So having a good signal-to-noise ratio is important, because if your noise floor is bigger than the signal, then you're not able to measure anymore. And usually we use dry electrodes because it's comfortable and fast to set up. You're always losing a bit compared to gelled electrodes, but if you have a good environment and you prepare them well, you can measure identical signals compared to gelled electrodes.
Then Arthur is asking a little bit similar, what's the difference between dry and wet electrodes?
One of the major differences is the impedance, and with the gel you also have a buffer. So if your electrode is shifting a bit, the gel acts like a buffer, and you are remaining good signal under more conditions. But as said, if you have good conditions, then you can measure identical signals.
Sadim is asking, can we get the data of the unicorn to a computer with Windows or Linux, offline or online?
We have all kind of software tools. For Linux, we have an API on GitHub. For Windows, we provide it via the Unicorn suite. So these are the two main systems where we support the Unicorn right now. And all of these APIs are real-time capable. So you can get data within a couple of milliseconds. And that's important to do experiments where you're flashing something and at the same time you want to look at the EG, and you have to synchronize the flashing event with the EG perfectly.
Subashini is asking which filter is optimal for noise and other artifacts.
There's no easy question. It always depends a bit. So there are all kind of different artifacts and all kind of different filter strategies. But commonly for EG, we use the power line hum. So if you're in the US, that's 60 Hertz notch filter, and in Europe, it's a 50 Hertz notch filter. So you have to check your geolocation and select the correct one. And we remove very low frequency noise with a high pass filter and very high frequency noise with a low pass filter.
And somebody's asking, can you have hair to use these EEG sensors?
Yeah, sure. Like the button is also having hair underneath. And for sure, it's important that the electrodes somehow touch the skin. And if you don't get a good contact, then gel also might help. But in general, it doesn't matter if you have hair or not. You just have to check that you're preparing your setup well.
Daria is asking, what did you change to remove the artifacts? Not just by just adding jails. So what else can you do to remove artifacts?
Like first of all, the mechanical setup and the hardware setup is the most important. You can, with all these software and filter strategies, you can recover some portion, but if you're not even measuring it, that's the worst. So you always should check that you're having a good hardware setup with connection to the skin of your electrodes. And then by software, you can add filters, IIR or FIR filters. The Oscar is using some more complex strategies. So you always have to think about the signals you want to measure, the artifacts that are destroying your signals there, and find some strategies for that application.
How long can you record with electrode jail setup without refilling is a question from Lino.
We tested around 6 hours plus, but after some times it's becoming dry, and then you just have to update it a bit.
And somebody says asking, can you explain neuromodulation?
I don't have a quick answer for that one right now.
Maybe I can answer that. So neuromodulation is a closed loop system. This means you are measuring, for example, from the brain or from the cortex with invasive or non-invasive technology. Then you are triggering or controlling a feedback device. This can be, for example, intracranial brain stimulation. This can be functional electrical stimulation of muscles or nerves. This can be transcranial magnetic stimulation. Important is that you're closing the feedback loop. A robotic system, for example, is also a neuromodulation system. You can see the robot, how it's moving. This is influencing the EEG data. So it's enhancing, for example, the oscillations because you're seeing the robot moving, and this improves accuracy. Then Lorenzo is asking, why is the training needed? What happens without training?
Yeah, like in our applications, we're using this closed loop system where we're also training some AI or machine learning algorithms, and they always need training data in order to perform well, and therefore you want to check your experiment, then extract the features for your AI algorithm. And then you need some training data in order to being able to apply something in real time. So therefore we have this training and application phase. You can also create BCIs without training, but they're usually not performing that well or stable. That's why we have training and application phases.
Adam is asking, which algorithm do you use for the blonded check?
Like I can't tell you the algorithm exactly, but there are the default strategies that shown before, like filtering for preconditioning, and then we have a classifier for classifying their different kind of algorithms for similar problems.
Then Reza is asking, what's the price of a unicorn hybrid black?
I think it's a little above 1,000 euros at the moment, 1,100 or something like that.
Yunshan is asking, can we use g.tec with other EEG devices or only with g.tec products?
g.tec is only working with g.tec devices at the moment.
And with that, we reached actually exactly 10 o'clock. Thank you very much, Martin, for the nice demo, and Sebastian for being our, subject. Very nice.